Welcome to the second video about hematocrit. This video will be an overview about the different methods used for hematocrit testing. We have two general ways in measuring hematocrit. We have the manual methods and the automated methods. The rule of three will also be discussed, but this is not considered as a method of measurement, but is a means of counter-checking if either the manual or the automated method were able to give the correct result. The manual method uses direct techniques in measuring hematocrit, while the automated and the rule of three uses indirect techniques. Examples of manual methods would be microhematocrit methods and macrohematocrit methods. Automated machines give computed or calculated means of getting a hematocrit, and the rule of three will be using a basic equation. Let's start with the first method, which is the manual method. And under this method, we have two types. We have the macro hematocrit method and the micro hematocrit method. Now, between these two types of method, macro hematocrit methods are no longer performed in the laboratory. We now use the micro hematocrit method for manual testing. But since this video is intended for students who will take the licensure examination, it's important that we still discuss some details about the macro hematocrit method. The general description given to macro hematocrit methods are as follows. They are considered obsolete in laboratory testing, and they are rarely used, usually for research purposes or for historical background. They are also time-consuming and require large amounts of blood. Another disadvantage of macro hematocrit methods is that they contain a higher percentage of trapped plasma that may lead to false results. The macrohematocrit method has 5 sub-methods, and we will discuss or differentiate these macro methods according to the specimen needed, the anticoagulant to be used, the test tube required, and the procedure to be followed. The first one is the most famous one, which is the Wintrobe and Landsberg method that uses whole blood, a double oxalate for an anticoagulant, a Wintrobe tube, and the procedure is to centrifuge the Wintrobe tube at 3,500 RPM for 30 minutes. The second macro method is the Van Allen method. This method uses capillary blood instead of whole blood. Anticoagulant to be used is 1.6 sodium oxalate, which is in distilled water. The test tube for this method is called the modified Van Allen hematocrit test tube. The procedure is to centrifuge this test tube in 2,500 RPM for 15 to 30 minutes. The third method is called the Hayden's modification method, and like the Wintrobe method, this also uses whole blood. And like the Van Allen method, it uses sodium oxalate 1.1%. The test tube to be used for this method is called a calibrated tube and should be centrifuged for 3,000 RPM, 20 minutes. The fourth one is called the Sanford-Magath method, which also uses whole blood and also uses sodium oxalate, but this time at 1.3%. The test tube used here is a calibrated tube with a funnel-like mouth. This should be centrifuged at the highest speed for 15 minutes. And the last macro method is called the Bray method, which also uses whole blood, and this time anticoagulant to be used should be heparin. The test tube is called the Bray's tube, and this should just be let alone standing for 1 hour. These are all the different types of macro methods. To make memorization easy, let's differentiate these 5 different methods. So out of the 5 methods, only the Van Allen method uses capillary blood. Out of the 5 methods, 3 of them will be using sodium oxalate, but only differing in the percentage that is needed. Out of the 5, only the Sanford and Magath method has a tube with a funnel-like mouth, And out of the 5 methods, only Bray method does not involve centrifugation. The test tubes in each of the methods are responsible for the method's uniqueness. So, on the next slides, we will be differentiating the different test tubes used in the different methods. The first tube to be described under the macro methods would be the Wintrobe tube, used for the Wintrobe and Landsberg method. This tube has a total length of 11.5 centimeters with a bore diameter of 2.5 to 3 millimeters. The top portion is open while the bottom end of this test tube is closed. This tube is also calibrated on both sides, the left side and the right side. The left side is used for the ESR determination, with the top increment being 0 and the bottom being 100 millimeter. The right side is the one used for hematocrit reading, and this time the top portion is 10 centimeters or 100 millimeters and the bottom is 0. The Van Allen method uses the modified Van Allen hematocrit tube, and this test tube has a bulb at the top portion and is calibrated from 1 to 10 centimeters or 10 to 100 millimeters. The next tubes to be described would be the ones used for the Sanford-Magath method and the Bray method. For the Sanford-Magath method, it is described to have a funnel-like mouth with a 6 ml capacity. This is only 5 inches long with 1 millimeter per division. The Bray tube, on the other hand, only has 5 ml volume capacity, also with a 1 millimeter division. This time it is calibrated from 10 to 50 millimeter. It has some similarities with the Wintrobe tube, which is calibrated on both sides, but this time the Bray tube has a flat bottom. The second manual method is the microhematocrit, which is also known as the Adams method. The microhematocrit is considered as the next best procedure after the automated analyzers. Tubes to be used for the microhematocrit method would be the capillary tube, and specimen may either come from a finger prick or a venipuncture using an anticoagulated test tube. If a specimen to be used is from a finger prick, the capillary tube to be used is the heparinized tube, since the finger prick does not have an anticoagulant yet. But if the specimen comes from a venipuncture with an anticoagulant, then a non-heparinized capillary tube should be used. Let's talk about the capillary tube a little bit more. The length of a capillary tube is from 7 to 7.5 centimeters or 70 to 75 millimeter. The bore, or the opening of the capillary tube, has a diameter of 1 millimeter, and it has a capacity to hold 0.05 ml of blood. The capillary tube has one end with a colored ring and the other end with none. And the end with no colored ring is where the blood should enter, because the side with the colored ring will be used for sealing the capillary tube. The clay seal should only be 4 to 6 millimeters long. And when we fill the capillary tube for hematocrit testing, we should only fill it up to 3/4 full or up to 5 centimeters. The automated methods have replaced the manual methods since the 1890s, and one of those tests that have been replaced by the automated method is, of course, the hematocrit method, and this is considered to be more accurate and more precise compared to the manual method. Let's give examples of different automated analyzers that test for the hematocrit together with their principles. So the Beckman-Coulter counter, the Celdine from Abbott, and Siemens Advia all have the same principle where the hematocrit count is computed using the red blood cell count and the mean cell volume. So red blood cell is multiplied to the mean cell volume, or MCV, and divided by 10. That would be the hematocrit count. The Sysmex, on the other hand, has a different principle which uses the cumulative RBC pulse height detection. We are now down to our last topic for this video, and this is the rule of 3. This rule is only applicable for normal red blood cells. So that means the red blood cell should be normocytic and normochromic, normal size and normal colored red blood cell. We do this so that we can check for the validity of the hematocrit result given by either the automated method or the manual method. When we perform the rule of 3, we need to have either the red blood cell count or the hemoglobin count. If a red blood cell count is given, we just multiply it by 3, hence the name rule of 3, so we can have the hemoglobin count. And if the hemoglobin count is given, we also multiply this by 3 so that we can get the hematocrit in percent. So why is this rule of 3 applicable? Because by principle, the hematocrit should be 3 times more than the value of hemoglobin. So if we have hemoglobin, if we multiply it by 3, we can therefore have a good estimate of the hematocrit value. Again, the rule of 3 is only for normal blood specimens, so that means it only applies to normocytic and normochromic red blood cells. It should become a habit for the medical technologist or the analyst to multiply the hemoglobin by 3 mentally so that we can find any discrepancy in the values given, so that if there are any errors, we can easily identify or find these errors. Let's give examples of how we can use the rule of 3 to verify if the automated or the manual result is acceptable or not acceptable. So this is, for example, one specimen. So this is sample 1, and it was processed through a manual method. And the results are an RBC count of 5, a hemoglobin of 15, and a hematocrit of 45. Now, to double check if these results are acceptable or not, we can use the rule of 3. So if we multiply the red blood cell count of 5 by 3, then we can get the hemoglobin value, which is 15 grams per deciliter. And if we multiply this again by 3, then we should get the hematocrit value of 45. Now, as we can see, the result from the manual method and the rule of 3 are the same, and for the hematocrit, they are the same as well. So that means the results are acceptable. Let's try another sample. So this is your sample number 2. And this was also performed using a manual method, and the results are an RBC count of 6, a hemoglobin of 11, and a hematocrit of 37. So to check if these are acceptable or not, we use the rule of 3. So an RBC count 6 multiplied to 3, we will get the hemoglobin of 18 grams per deciliter. And if we multiply this again by 3, we should get the hematocrit value of 54. But this time, as you can see, the manual result and the rule of 3 result do not comply with each other. The hematocrit value and the rule of 3 value do not comply with each other as well. So that means these results are not acceptable. Let's have another example. So this is sample number 3 with the following results. Red blood cell count of 6, hemoglobin of 16 grams per deciliter, and hematocrit of 57%. Using the rule of 3, I want you to find out if these results are acceptable or not acceptable. I will give you 15 seconds to find out. All right, so time is up. Let's use the rule of 3. Red blood cell count 6 multiplied to 3. We will have a hemoglobin value of 18 grams per deciliter. And if we multiply this again by 3, we will have 54% for hematocrit. So are these values acceptable or not acceptable? The answer is that they are acceptable. So why are they acceptable when the values are not the same? The reason for that is because they still fall inside the reference range of the rule of 3. For hemoglobin, we can add or subtract 1.5 to 2 grams per deciliter from the expected value, and for hematocrit, we can add or subtract 3% from the expected value. So therefore, the acceptable range for hemoglobin is 16 to 20 grams per deciliter, while for hematocrit, the acceptable range is 51 to 57%. Now, since our result is 16, which is still inside the reference range, the hemoglobin value is then acceptable, and the hematocrit of 57% is still within the acceptable range. This makes the hematocrit value acceptable as well. So what do we do if the values do not agree with each other? First, we have to examine the blood film or the blood smear of the patient. And then from there, we have to find out if there are any causes of a false increase or a false decrease of the hemoglobin or hematocrit value. Let's now give examples on what we should do when we find any discrepancies. So let's say we have a hemoglobin value of 9 and a hematocrit of 32%. If we use the rule of 3, ideally when we multiply 9 by 3, we should get 27%, but our result is 32. Then we need to consider the reference range. It's plus minus 3 for hematocrit, so the acceptable range is 24 to 30%. So 32 is outside the acceptable range, so that means that initially this would be not acceptable. But before we really rule the results out, we need to do some investigation. So first we need to look at the blood film or the blood smear of the patient. Now there are two possible results, one being the blood smear normal and the other one being abnormal. Let's say that the blood film shows hypochromic, so that means the red blood cells lack in color, and that they are microcytic, so that means they are small red blood cells. That means the rule of 3 cannot be applied, because remember, the rule of 3 is only applicable for normocytic and normochromic red blood cells. If this is the case, then that means that the hemoglobin and hematocrit should be released as is. But let's say, for example, that the smear shows normal red blood cells, normocytic and normochromic, so then that means the rule of 3 can be used and say that the 32% hematocrit might be wrong. So what do you do next? You have to investigate on the possible causes of a false increase or a false decrease of the hematocrit. So in this case, it is a false increase of hematocrit. This is our last example. So for example, we have a hemoglobin of 15 and a hematocrit of 36. The rule of 3 would say that the hematocrit value should be 45% with an acceptable range of 42 to 48%. So initially, this would mean that this hematocrit value is not acceptable. But we should first check the smear or the blood film of the patient so we can have an abnormal result or a normal result. If in case the blood smear shows hypochromic and microcytic red blood cells, that means the rule of 3 cannot be applied, so these results can be released. But if, for example, the blood smear shows normocytic and normochromic red blood cells, then we have to do further investigation on what might have caused the discrepancy of the hemoglobin and the hematocrit with the rule of 3. So let's say, for example, after investigation, we found out that the specimen is lipemic, and lipemia in the specimen would cause a false increase in hemoglobin. So that means our hemoglobin is falsely high. That's why when we computed with the rule of 3, we have a high expected value. So what we have to do now is we have to correct the value of the hemoglobin before we use the rule of 3. But what if there are unexplained discrepancy even after investigation? What else can you do? Well, you can run the samples before and after the specimen in question. Now, if these samples do not conform with the rule of 3, then there should be further investigations so that the problem can be found. What you can do still is you can run a control sample. So this is your quality control to check if the machine is working properly. If the machine is out of control, then that means you should fix your machine first. It is the one causing the problems. But if it is in control, then we can say that maybe that this is just a random error. And that is it for the overview of the different hematocrit methods. Thank you very much for watching. Make sure that you watch the videos of the different procedures on how to perform the hematocrit test.